So welcome again, everybody. This is the 2024 SnapBean conference, and our first presentation is going to be with identification management from Calvin Odero. And the stage is yours.
Okay, can you hear me well?
We can.
Okay. Good morning, everyone, and thank you for taking your time to attend the SnapBean meeting. I'll be giving an overview of some of the common weeds that are associated with the crop and some management tips or issues that we can look at when when managing some of these weeds. SnapBean, as you're all aware, is a very short season crop that is very sensitive to weed competition. And SnapBean plants will compete poorly against weeds. As you know, weeds will compete with bean plants for water, nutrients, and sunlight, and thereby decrease crop yield and quality. So there are several studies that have documented yield losses in snap beans and similar species such as dry bean, or even up to 80% or more from weed interference, depending on the weed species composition or density. So some of the common weeds, put pictures here of weed seedlings because it's important to know how to identify weeds at this stage, because this is when they're most vulnerable or susceptible to control with most of the herbicides that we have. So several annual broadleaf weeds species are a problem in our bean fields, and these would include several Amaranthus species. The first one I have here is spiny amaranth, which usually has an erect stem. It's going to be red on the stem with a pair of spines at the node. And then the next one would be smooth pigweed, which will almost look similar, but it's not going to have any spines on it. We'll have a little bit of a notch tip. And common lamb squatters, the main key to identifying it is that it has a gray mealy coating on the leaf, and it typically has an evenly toothed margin. And then the American nightshade has dark green leaves. It's typically covered in hairs, and then the stems are usually going to be purplish and hairy. And then common ragweed, as you can see, the leaves are very deeply lobed. It's also covered in hair. The stems are going to be hairy. And as you can see, the stem is usually also purplish in color. And then common purslane, these will have thick and succulent leaves that are spatula shaped. It doesn't have a leaf stalk or a petiole. And all these weeds will cause, I would say major to moderate yield or quality losses when they are not adequately controlled in a timely manner. There's another amaranth here species that you can also commonly see in beans. It's more prostrate. It's called livid amaranth. It has no spines. It's going to be prostrate, as I said, and then it has a characteristic notch on the leaf tip. And then common ragweed, it depends. Typically more on the mark soils. Maybe you can see on the beans. For ragweed parthenium, the first leaves, the plant will have a rosette type of growth initially, and the leaves will have very fine hairs, and subsequent leaves, as you can see, will be really, really deeply lobed. And then we do have the nutsedges. Usually yellow nutsedge and purple nutsedge, which are perennial weeds, are the ones that can be found in bean fields. And these will typically form dense infestations when they are not controlled. So all the broadleaf weeds that I mentioned are all annual weeds. So none of them is a perennial weed. And yellow nutsedge stems, can grow up to 3 feet tall, while purple nutsedge doesn't grow as tall, but can reach over 1 foot, say typically about 16 inches. And then the main difference between the two, the main difference between the two is that yellow nutsedge leaves gradually taper to a sharp point. That's the picture here. While purple nutsedge leaves abruptly taper to a sharp point. And the easy way to tell the difference between the two is when you pull the plant, yellow nutsedge tubers, you'll find them growing at the end of the rhizomes, as shown in this picture here. while for purple nutsedge, the tubers will grow in chain. So if you pull any sage that is in chain, that you'll know that it's a purple nutsedge. And these two will also cause major to moderate yield losses depending on the level of infestation. For grasses, for the bean crop, grasses are relatively easy to control with some of the graminicides, Select or Post. Does a very good job in managing them. But some of the common ones that you see in beans, depending on whether you're on mark or on sand, fall panicum. The main way to identify is that it has a ligule that is hairy, and then the leaf sheath and blade, typically on the lower side, will be hairy, but it usually typically becomes less hairy as it ages. And then the southern crabgrass is going to be very, very hairy. And if you look at the ligule is going to be a jagged membrane, a very, very huge membrane. And then goosegrass is typically very smooth. It's very flattened if you look at the stem. And it will have also a tooth membrane that is very uneven. On some of the sandland areas, you can come across the sandbars. When they're mature, they have the fruit is more of a bar that sticks on clothes or anything, or even can stick on animals. But if you look at it, the plant is hairless. It will be compressed. You touch the leaves, will have a very rough leaf blade, and then the base of the stem is always tint red. Another weed you can come across would be crowfoot grass, which is kind of smooth, but it'll have long stiff hairs on the leaf margin. And the ligule is a membrane, usually with a fringe of hairs at the top. And then the Bermuda grass, which is a perennial weed, you can come across that too. The ligule for Bermuda grass is going to be hairy, but the plant itself is going to be smooth and kind of rolled in a bud. So here is a graph showing, the critical period of weed control, snap bean on mark soil. And what we know is that correct timing of weed control measures in snap bean is important in order to optimize yield, given the crop's short growth cycle. So the critical period of weed control is that time interval in the crop's growth cycle during which weed control measures must be undertaken to prevent any yield losses. And these yield loss numbers are more of kind of, I would say, arbitrary. They can range typically from 2.5 to 10%. So it depends just on the yield loss level that a grower is willing to accept. So for beans grown on mark soil, the critical period we control to avoid a 2.5% yield loss is, will start from the VC stage, which is the cotyledon or the unifoliate leaf, all the way until the mid pod set, or when 50% of the pods have reached their maximum length, which is almost the entire growing season. And to avoid a 5% yield loss, the weeds have to be, the fields have to be controlled, free of weeds from the first to the second trifoliate leaf, all the way until the mid flower or early pod set, or when 50% of flowers are open, and some of the pods have reached the maximum length. And then for a 10% yield loss, you have to control weeds from the second trifoliate leaf all the way until early flowering. So these intervals basically show us that weed control in snap bean is required throughout most of the growing season to maximize yield and minimize the effects of weed interference or weed competition. Therefore, when looking at weed control, we can say that weed control, you can use residual pre-emergence herbicides, maybe in combination with post-emergence herbicides or tillage, in order to provide weed control throughout most of the season to prevent any yield losses that you may end up with. So how do you plan for an effective weed control program where you end up, moving from a field, this is not even very weedy, to a field that is as clean as the one on my right side? So to develop an effective weed control strategy, it's always important to consider what the weed species that are present in the field. Consider things like if you have used a cover crop, how are you gonna get rid of the cover crop? Things like pre-plant tillage, the herbicides that you're going to use, and even herbicide incorporation if they are going to be pre-emergence herbicides. And then also ensure that you conduct accurate weed identification, which is very, very critical. It's very important because it will enable you to have the most effective and economical control program once you know what the weed spectrum is in the field based on scouting or just based on the previous crops that had been planted in the field prior to your current crop that your bean crop that you're going to plant. And then you have to ensure that you use an integrated approach when it comes to management. So mechanical control, tillage, cultivation helps a lot. And of course, cultural control, things like crop rotation, using cover crops, correct fertilizer placement. And we mostly rely heavily on chemical control or herbicides, but using herbicides, it's good to use herbicides in combination with tillage or in order to have a more effective control of some of the weed species that might be present in the field. So this is a chart that shows different herbicides that can be used for snap beans, and each has different efficacy depending on the weed species. So for pre-emergence herbicides, I know in our area people typically use dual magnum, which is S-metolachlor, which is generally good for mostly for grass control, but can have some fair to good activity on a few broadleaf weeds. Other pre-emergence herbicides would include, Treflan, Prowl. Treflan and Prowl. Treflan is the same group as Prowl. It's one of the yellow herbicides too. Pursuit, Reflex, which is also used post-emergence, and Eptam. And then for pre-emergence, you can still, for post-emergence, you can still use Pursuit, Bazagran, and Reflex are the most commonly used in our area. You can use Sandia or mostly for nut control. And then for grass control, we've got, Assure, but I know most people use, majorly Post, a few people use Select Max. And then a combination of Pursuit or Bazagran. I don't know if anyone uses it in our area, or you can also even use a combination of Reflex and Bazagran. So for pre-emergence herbicides, I wanted to put this because I'll show you some work that I started looking at one of the pre-emergence herbicides that I listed here, which is Eptam. Eptam is a herbicide that can be used pre for broad spectrum weed control. It has activity on grasses and some broadleaf weeds, plus some fair activity, particularly on yellow nut sedges. So pre-emergence herbicide application, of course, is a proactive and effective strategy for weed management that offers numerous benefits, including early weed control, efficient use of resources, and can also result in cost savings if you don't end up using any other post-emergence applications or herbicides. So pre-emergence herbicides prevent germinated weed seedlings from becoming established either by inhibiting the growth of the root, the shoot, or both. And pre-emergence herbicides must be mechanically incorporated into the soil to control these germinating weed seedlings. Pre-emergence herbicides generally require rainfall. If they cannot be mechanically incorporated, then they require either rainfall or irrigation after application, but prior to weed germination to have them incorporated. So as I mentioned, some of the common pre-herbicides were Dual, Eptam, Prowl, Treflan, or Reflex. And Eptam, or EPTC, it's a thiocarbamate. It's a very long-chain fatty acid inhibitor. It's in the same group as Dual. And its use rate is from 2¼ pints to 4½ pints per acre, which is 2 to 4 pounds of active ingredient per acre. So it will be absorbed onto soil, and absorption increases as organic matter content increases. And with regard to leaching, the leaching will decrease as clay or organic matter content in the soil increase. And it's going to be readily lost from the soil surface when it's moist at the time of application if it's not incorporated. So one thing with the EPTAM is that you really have to incorporate it mostly mechanically to make it to be very effective. So I wanted to share with you some a study that I did this last fall on a sandy soil. So we conducted a field study to look at the efficacy of different pre-herbicides. The goal was to see how EPTAM would work in relation to the other commonly used pre-herbicides. So we had DOL at 1.33 pints per acre, Reflex at 1 pint and 1.5 pints, and then EPTAM at the 2 rates, 2¼ and 1.5 pints. But as you can see from this graph, the level of weed control was not more than 50% for any of the herbicides. The level of weed control was not good because we didn't have any incorporation. I tried to incorporate because the fields were bedded right after planting. Then I went in and just applied the pre's. I tried incorporating the herbicide by using a rake, but that didn't work because, you know, for mechanical incorporation, you need to incorporate the herbicides, to about 4 inches shortly after application to avoid, especially for EPTAM, to avoid volatilization or photodegradation losses, especially if the soils are very moist. And then these would then be followed by planting. So I don't know if there are people, if there are growers who have a setup of a tractor or tillage tool that you can have with tanks for incorporation that can be used, kind of mounted on the same thing, and then after that you plant right after. So I would like to look at this further using mechanical pre-plant incorporation, especially for EPTAM, followed by planting in an ideal world, to see what the efficacy is going to be on a sandy soils. Some of the weed species, as you can see from this figure, were mostly, I had lamb squatters, some smooth figweed, smartweed, and some southern crabgrasses. So anyway, I'll continue to expand more on that, to look at just some of the pre-emergence herbicides and look at what their efficacies are going to be over the course of hopefully this coming season. And to end my talk, for herbicide use, always remember to scout your fields, make sure you identify weeds correctly. Then with that, you select your appropriate herbicides that you ensure that you have the effective, and ensure that you also do proper equipment calibration, so that way when you're applying the herbicide, you apply them at the right rate. And always make sure for chemical control, do it right, because as I mentioned before, have proper herbicides, proper herbicide placement, proper time of application, and proper manner of application, and always make sure that you follow the label. So with that, thank you so much. I don't know if I have any time for questions.
No, unfortunately, we don't have, but I know you are going to share your slides, and also we are recording this, so you're going to, all of you you're going to be available. You are going to be able to see the recording of this. So thank you, Calvin. And
all right, thank you, Anne.
We have your contact information, so if anybody has any question, that's another way to contact him too.
Okay.
Thank you again.
All right, thank you.
Have a good graduation.
All right, thanks.
Thank you.
Thank you.
All right, just can unshare your screen probably. Yeah, okay. I'm gonna try to share this one. And so our next speaker is going to be Dr. Katya Javier. She's here at the research and Everglades Research and Education Center, and she's going to talk about SNAP bean disease management and ID. what we've seen this year, actually. Thank you.
Hi, good morning. Thank you, Anna, for the introduction. I'm glad to be here and to talk a little bit about disease identification management for snap beans. So I don't have any project on snap beans yet, and I would like to have your ideas later. Maybe at lunchtime, if you guys have ideas, let's talk, because I'm planning to write a proposal on snap beans. But anyway, let me get started because I just have 15 minutes. So since I don't have a project and data to show today, what I did to put this talk together was to based on a survey that I did 2 years ago. And also I got some reports, and Anna helped me with that, to contact Romina Gaziz in TRAC and Pamela Roberts in Southwest Research and Education Center. And they sent me some information. So based on that, we can see that Pythium and Rhizoctonia, they sometimes they're kind of together on the reports. And so this is, I would consider the most important one, and I'll be talking about them. And then the viruses, they're also important. So I'll talk about them too. White mold, it's important. This year we saw, I got some samples here too with white mold. So I'll consider that important one. And then after that, Alternaria and rust. Okay, so I'll be talking about those. So first, I'll talk about Pythium and Rhizoctonia. Okay, but before I get to that, so there are some pathogens that they call soil-borne pathogens. So that means they can live in the soil, crop debris, and they will infect the root system. And from there, they will cause disease. Now, there are some other pathogens that come through the air. So those are called airborne pathogens. So they will infect the leaves, and they can cause spots, blights, and so on. So first I'll talk about the soil-borne pathogens like Rhizoctonia and Pythium. So they infect, as I said, the root system, and then from there, they move upwards on the plants. So they can block the vascular system. So one of the symptoms is wilt. And then they can cause that discoloration on the root system. So if you can cut it, you are going to see discoloration. So those are the symptoms. And they can eventually kill the plant. So the plant will die. So you don't have any yield from that plant. So that's really bad. Now for this disease, root rot, the management strategy that was listed that I could find was chemical control. Now, it's important to keep in mind that actually Pythium is not a true fungi, okay? So it needs a specific fungicide to control it. But Rhizoctonia is a fungi, so it can be treated with, for example, Quadris. So if you send your sample here and I tell you it has Pythium and Rhizoctonia, probably you wanna apply both. Now, if I tell you, oh, I just identified Rhizoctonia on your sample, and then you can spray or treat it with only Quadris, okay? So that's really important to keep in mind, especially if we find Pythium on the sample, because then you need a specific one. And that's true for other samples too, like sod. I got a lot of sod this year. And sometimes I would identify Pythium and Rhizoctonia, but sometimes just one of them, okay? So that's important in terms of management strategies. Now, another pathogen, Sclerotinia sclerotiorium, that causes the disease white mold. So if you're in the field and you see something like that, that's easy to do the diagnosis, right? But sometimes what you see is not exactly that. This year, Glades Crop Care, Dave brought me a sample in a bag, and I could see some pods, but they didn't have any growth like that, like white mold. I could see a pod like that. And the symptom that it has was like watery soft rot. And I was like, what is that? You know, under the microscope, I could see the hyphae and I could tell, oh, white mold, because the hyphae is thick and all of that. So for me, it was kind of easy to do the identification, but out in the field, sometimes you see that and you don't know exactly how that's going to evolve. So if you have questions, just bring the samples and we are going to look under the microscope and help you with the diagnosis. But after some days, if I just incubate the samples, I'm also going to see that white cottony mycelium on the sample. And another thing is that this pathogen is going to produce what's called sclerotia in the soil or in a sample like that in here. It's where it's starting to formate that. It's like, it looked like a bean, you know, like black beans. It looked like that. It's have like irregular shape. And what's interesting about this pathogen or this structure that the pathogen produces is that it's going to survive in the soil for many, many years. And then this structure can actually germinate. And from that, the spores can be disseminated. And then it can become an airborne pathogen because those spores can disseminate and infect the leaves and cause symptoms on the leaves, okay, or pods, because when it gets there, it starts to cause the disease again. So there is also some chemistries that can be used against this pathogen, and they are listed on the Vegetable Production Handbook of Florida. So that's why I'm not going to read it. Anna is going to make this presentation available, and also you have all this information on this book that's also free, right? It's for free, and we have it available here, so you can just get it later. So I think that's all about this disease. Did I tell this? Oh, yeah, that's, I know there was something else, but it's also important that if you have high infestation of or infection of this pathogen in a field, it's important to, and I think most of the growers already do it here, is to do the tillage of that field and also to flood that field. There were some studies that were done here in this area, and they observed that after flooding it, it would reduce this sclerotia in the soil because it would kind of rot it. Now, another wind-borne pathogen is rust, and this rust is specific for beans. Let's say the rust that infects sugarcane cannot infect beans, and the ones that's on beans cannot infect sugarcane. So it's what they call host specific. So this disease can reduce yields by 30%, and the ideal temperature is 60 to 72 degrees Fahrenheit. And there are some host resistance or tolerant varieties that you can use against this disease. And it's important, especially for like organic growers, because they don't have many strategies that they can use against this one, because the main strategy is actually to spray with fungicides. So if we start with some varieties that have some degree of tolerance, maybe that can help some. So here are the chemistries that are labeled for this, to control this disease, if you're a conventional grower. Now for the Alternaria pod spot or leaf spot, is caused by Alternaria alternata. And you can see, I'm trying to show here on these pods, you can see the tiny dots or spots on the pods. They have this black color. And if we look, if they get a little bit bigger, we can see these zonate or target-like lesions. That's very characteristic for Alternaria. But if you have questions, you can bring the samples and we can help you with the diagnosis. And there are several cases where this disease has been correlated with injuries from spider mites, and also it's correlated with nutrient stress. So that's important to know because if it's correlated with spider mites or nutrient stress, if you think about control strategies, you have to think about, you know, a good nutrient balance for those plants and also control mites infestations if you have, and that's very common on snap beans, right? And for chemical control, it's recommended to do two applications with Quadris. Now talking about the viroses, in here we have listed two, the bean golden mosaic virus and the bean common mosaic virus. And the symptoms will be similar. Actually, viroses in general, you think about curly leaves, and usually they curl downward, and the plants get stunted. And if that occurs like pre-bloom, that infection can lead to losses by up to 90%. So that's very important. And then if we talk about viruses, the seeds can be infested with that virus. But then if the plants, if you have that source of inoculum, what happens is that if we have the vectors that can be whiteflies or aphids, they are going to feed on that plant that's infected. And then from there, it starts to transmit the disease and to disseminate that disease in the field. And then that can be a big problem, especially when the temperatures are high, because that's going to favor the insect population. I'm not an entomologist, but I kind of know that. And then, and then from that, what happens is that you can have big losses, right? That can be up to 90%. So what's important here is that you start with certified seeds and you control the insect population. There is a threshold to control that insect population. And that's why scouting the fields is very important. As I said, I'm not an entomologist, so I cannot tell you exactly what the threshold is, but you can talk to our entomologists in here and they can help you. And also host resistance is available for some of these viruses. Another one that's important is the bean red node virus. I always think about red nose. I don't know why, but it's bean red node virus. So, and the vectors here will be two species of thrips. And if you see the agenda, they will be talking about thrips after this presentation. So you can learn a lot about that. And there were like there was a severe outbreak in 2020 here with some fields that they reported losses by up to 100%. So that was a very important disease in 2020. In terms of management, it's recommended to use certified seeds and also to control those vectors that are the thrips. Now, what about management, right, for all those diseases? So when you talk about managing pathogens, managing insects, you cannot just think about one tool in the toolbox. You have to think about several things that you have to do to control these pathogens. So it's important to start clean. So it's important to start with certified seeds. It's important to do like site selection in a sense, like if you have for those soil-borne pathogens like Rhizoctonia, Pythium, white mold, if you have those diseases or pathogens in one block in your farm, you have to keep that in mind, because if you plant snap beans there again, most likely you're going to have problems and big losses for the next season. So it's important to plan your crop rotation well. In terms of chemical control for insects and for fungal diseases, it's important to rotate mode of actions. It's also very important to read the label and to apply the right dosage that's recommended, because you don't want to create resistance in that fungal or pathogen pathogen population or insect population. in this book, Vegetable Production Handbook of Florida, there is this table too, where there are some varieties or cultivars that are listed as the ones planted here. What I did was to read each one of these and see which resistance they kind of hold, right, against diseases and also heat. And I put this information together. So in here we have the cultivar and we have the resistance and the level of resistance actually that they have against virus and against fungi, against bacteria and the heat. So these slides will be available later, so you can have access to that. Or you can take a picture if you want to. And then in summary, in South Florida, seems that the diseases caused by fungi and virus are the most important problems on snap beans. And the disease management first depends on the correct identification of the problem. And then once you identify it, it's important to use an integrated management approach, including both cultural and chemical controls. And so you should adopt it in order to reduce the impact of the diseases on the snap bean production. With that, I would like to thank my lab and the growers and the crop consultants and all of you for paying attention today. Thank you.
Thank you, Katya. We are running a little bit behind, so if you have any questions to Katya, please find her during the break. And then also, like I said, it's going to be shared on our website. And also, if you need the slides, we can send it to you. So our next presenter is going to be Doug Sale. We switched a little bit, a few things around. Let me close this one. And then he's going to talk about overall thrips management in snap bean in South Florida. He's the entomologist down at the Tropical Research and Education Center. Thank you.
Thank you, Anna. Thank you, Anna. Thank you all. My name is Doug. I'm from Tropical Research and Education Center, Homestead, Florida. I work with the vegetable insect pest management. Before I start my talk, I'd like to thank Anna. She really helped me to prepare these slides and to organize this talk. Thank you, Anna, for your all helps. Today, I'll be talking about the snap bean, thrips management on snap bean. Snap bean as a whole, bean is an important crop in South Florida in Miami-Dade County. As a matter of fact, it is right now the #1 crop, vegetable crop. But we have a lot of problems. #1 problem is these thrips. We have to manage these thrips, otherwise it is difficult to harvest any bean. On snap beans, when I say thrips, we have two major thrips. One is melon thrips and another one is Asian bean thrips. Okay. You can see that the top one is Asian bean thrips and bottom one is melon thrips. Well, we have some thrips parvispanus too, but the number is very few on bean. It's mostly on other crops, and Anna will talk about that, I believe. So I will, in all my talk, I'll mention about these two thrips. Here, this slide shows Asian bean thrips damage. You can see on, this is a different, it's not snap bean, it's another bean, green peas. And you can see the damage on the fruit, on the leaves. This is a melon thrips damage, very serious damage. This slide is old, but at the beginning, when it came, it used to defoliate the field, total defoliation. And you can see there, And there is only one treatment that save it. This is Carzol, which is not labeled for bean. Talking about abundance of these thrips on different crops and bean varieties. Here is this slide shows that this study was conducted in the greenhouse condition. And you can see that bean is the number one. Yeah. This is bean, you can see the most preferred host of Asian bean thrips. This is for Asian bean thrips. So these slides, eggplant, pepper, and tomato, we found very few. But the idea behind this slide is to let you know that when you plant bean nearby another bean, you may have infestation of these bean thrips, heavy infestation. So, but if you have other crops, the infestation will be less. Here shows different kind of bean varieties we grow in Miami-Dade County, and I'm showing you the preference of this or abundance of these bean thrips or Asian bean thrips on different cultivars of beans. Here it shows abundance of Asian bean thrips on all these cultivars. The bottom line shows you cultivars: snap bean, kidney bean, hyacinth bean, navy bean, then black eyed peas, and again snap bean, two varieties, Opus and Myrada. You can see that hyacinth bean, this is the most preferred one we found there. And the two bars in his set, one is based on the flower sample, one is based on the leaf sample. The blue one is based on the flower sample. It shows you, this slide shows you that Asian bean thrips abundance is very heavy on flowers. On leaves, if you sample leaf, you may make some mistake because you will not get the right information. So it's better to sample flower for this Asian bean thrips. Next slide is for melon thrips, totally different. You can see that blue color is based on the flower and the yellowish color based on the leaves. So in case of the Asian melon thrips, you see more abundant on the leaves than the flowers. So in this case, you have to, when you think about melon thrips, you have to sample leaves. This is a Franklinella occidentalis. We get very few, and you can get them on both by sampling both flower and leaves. So here, as I mentioned that the Asian bean thrips on flower, this is a slide. It shows the abundance of Asian bean thrips on different parts of the bean plants. You can see that this is your flower, the next one is leaf, and the next one is pods. You see most of them on the flower. And I conducted this study in 3 fields. That's why there are 3 bars. In each field, we found highest number on flower. Now, dial pattern of distribution. I'll show you this slide just to let you know that what time we should spray our field. This shows the abundance of Asian bean thrips at different time of the day. You can see that. Yeah. So the bottom 2 slides, 2 bars, one is for at 10 a.m. and another is at 12 p.m. And highest number, peak activity of these thrips were observed before on or before 12. So best time to spray for managing this thrip is in the morning time, 8 to 12 p.m. Yeah. This shows the height distribution. This is, I did this work only to know how high they fly. And we found that and we conducted this study by using yellow sticky trap in the field at different height. And we found that most of them below 6 feet, mostly 2 feet is their flight activity in the field. So the thing we found that if you put any barrier around your field, you may limit the abundance of these thrips at the beginning of the season. It will be helpful. Then I'll be talking about management. In management, I used neonicotinoid insecticide, diamide, carbamate, pyrethroid, and biological insecticides. That will be one study. In my second study, I used Torac Radiant program. Third study, I used Radiant, Plinazolin, and Torac comparison. Fourth study, I used Radiant, Transform, and Lanate. Materials method little bit. In all these studies, I sprayed this insecticide 4 times at weekly intervals. Then I collected the leaves from his treatment plot. I brought them and I washed those samples with 70% alcohol. And then I counted them under microscope. This is the study. I used all those insecticides, Spinotoram, which is Radiant, Torac, XRL, Movento, and all those insecticides you can see. On the last column, it shows the rate of each insecticide. Here this shows, excuse me, the Asian bean thrips, effect of those insecticides on Asian bean thrips. I will not read all those bars. You can see that first 2 bars, which is Radiant, Torac, and Lanate, they provided significant reduction of these thrips as in comparison to the control. You see the control bar is at the right-hand side. This shows melon thrips control. These are all adults, based on the adults. In case of melon thrips, the result is a little bit different. Here you can see that 2 bars, one is Asail and Shibanto. That Asail and Shibanto provided significant control along with the Hero. This 3 insecticide was effective in this study. We did not get a whole lot of good result by using Torac, Radiant, in case of melon thrips. This shows the larvae. Previous 2 slides, I showed the control of adults. Here it shows the larvae. In case of larvae, you can see that Radiant, Torac, and Lanate, it's almost like the same result like the first slide, which was for Asian bean thrips adults. So it tells us that most of the larvae belong to that Asian bean thrips. That's why we found exactly the same result. Radiant, Torac, and Lanate provided good control of these these larvae. Here, these slides together. I will not read this. I already talked about them. This is our Radiant and Torac program. Our treatments are Radiant by itself, Torac by itself, Torac, Radiant in rotation. And all those treatments are used weekly basis. And you can see that the first column, it shows the rate of its insecticide, and second 2 columns for adult control, and last 2 columns for larvae control. You can see that the adults were controlled significantly by Torac in combination with Induce and Torac Radiant. These 2 treatments provided good control of larvae. In case of larvae, only Torac itself provided significant reduction. The control number is at the bottom of the chart. This is on old slides, but I tried it 2021. Radiant, Lanate, Guthion, and Agromac. These are the treatments. And believe it or not, that all these treatments, they really significantly reduced, but Radiant did the best job. And Lanate, Guthion is an old insecticide. I tried to see how does it work now, and it performed really good. All these were done in the field on snap beans and applied weekly. This shows our Radiant Plinazolin comparison. Plinazolin is not yet labeled, but this trial was conducted for IR4, and we found that you can see that it shows Asian bean thrips control, melon thrips control, and larvae control. Asian bean thrips and larvae were significantly reduced by Plinazolin, both rates. One was 15.3, ounce, another was 20.5 ounce. Both rates significantly reduced this Asian bean thrips and larvae. We did not get significant result for melon thrips. Here, this one shows again Radiant Transform. Radiant, you know that this is the product this product is performing really very well in controlling all kind of thrips. Here we use Radiant, Transform, and Lanate. We had 2 rates of Lanate, and you can see as you compare with the untreated control that the result was really good, significantly reduction we observed in case of Radiant by itself. Transform did good job, and Lanate both rates significantly reduced. At the same time, all these insecticides like Torec was also good in these trials. Here, this one is for Asian bean thrips, adults. We see that only Lanate and Radiant significantly reduced these Asian bean thrips adults. Here is one slide I like to remind you that this is Orius insidiosus. This is very useful. This is our native predator, and it caused this revolutionary effect in reducing melon thrips and other thrips. We really ignored them by using a lot of insecticide, knowingly and unknowingly. As a result, Orius population goes down. When season is over, all these insects move to the woody area or non-farming area. And in those areas, Orius is there and they keep the population under balance. So in the field, we can use them and we should be careful about saving them. Thank you. If you have any question, I think I'm almost done. The time was?
We wrapped up. Oh, thank you, Dak. Thank you so much. All right. Again, if you have any questions, you can find Dak during the break. Thank you again. So, and the last presentation from my UFIFAS, it's going to be mine about Thrips parvispinus. Just let me pull this up. Okay. So, just quickly, Thrips parvispinus update. Would bean production be impacted? Hopefully everybody knows by then that Thrips parvispinus is an invasive polyphagous species, thrip species that affects the production of vegetables and ornamentals worldwide. It's reported feeding on over 40 different plant species from a variety of different crops. That's why is probably super important. Thrips parvispinus was first detected in the continental United States in Orange County in 2020. On ornamentals. And by early 2022, it made it down to Miami-Dade County, causing more and more problem to local ornamental growers and nursery growers. Later in the spring, it was recorded from a gardenia hedge up in Palm Beach County on Palm Beach Island. And this, by September, this population was a well-established population, and we heard that people had more and more hard time to control it. You can see that from the back, it's a really, really tiny thrips. So, well, anyway. In November 2022, this is when everything went sideways. It was reported in a commercial pepper fields for the first time in Palm Beach County. And this was the very first record of an established population in Florida in a vegetable field. And actually, that was in the first record in the continental United States from a vegetable field. So this map shows Thrips parvispinus surveys conducted by FDAC's DPI inspectors. And why? Because it's a regulated pest. It's still a regulated pest. The green crosses here represents a negative survey by FDAC's DPI, and the red dots, try to show it with this one. The red dots, they are positive survey, represent interceptions, and interceptions is in ornamental nurseries or in stock dealers. And why this is important and why relevant? Because to prevent further spread, FDAC's DPI implemented a quarantine. So what does it mean? That under this regulation, nurseries found with Thrips parvispinus are placed in stop sale and hold order. nurseries could not and still cannot move any infested plant materials until the pest has been eradicated. And the good news is that as of as of April 26, 25, we had no active quarantine. So the counties, when you see here with gray color on the East Coast mainly, this is where we have reports of Thrips parvispinus from the landscape, either from ornamentals or also from vegetable host in the ground. So that means that the thrips in these counties are established. Monroe County, Palm Beach County, you see all those counties up there. And Palm Beach County was the one actually where the most of the damage was recorded in commercial pepper field. As you can see here on the picture, parvispinus is a small thrips, very, very small, smaller than Asian bean thrips. So we heard about the bad thrips about 2 years ago. That was the Asian bean thrips. This year, the bad thrips is parvispinus. But you can see the females with the naked eye here. I circled them with a red color circle. And they have a unique color pattern, yellowish thorax and a dark abdomen. And the males are yellowish and smaller than the females. Here, just, I don't know if I can start this quick video. You can see the male, they are yellow. And then the females, they have this bicolor and they are feeding on a pepper plant. So this is what these thrips can do with small pepper plants when it's not controlled. It's important to notice when it's not controlled. You can see how the terminal leaves and the flower buds are completely deformed and destroyed. For the first look, it was somewhat looked like a heavy broad mite injury. The damage was so severe, pepper growers lost millions of dollars during this 2022 and 23 winter pepper production season. So should bean growers be concerned? Will Thrips parvispinus be the new Asian bean thrips? So what we've done and what we've been doing with Dr. Defen Mu and Julian Bilsland, we're conducting survey and monitoring thrips population in vegetable fields, mainly in Eastern Palm Beach County, where infestation were high during the 2022 and 23 season. So what we know that beside pepper, cucurbits can be infested too, especially when they are planted behind pepper in the same field. Here you can see the adults on the back of the leaves, on the side of the cucumber leaf and on squash flower. And fortunately, the injury on cucumber and squash seems to be minimal compared to what we could see or what we've seen on pepper with this level of infestation. We found parvispinus on eggplant on this farm too, but it did not cause significant damage, which is good. For snap bean, very, very low infestation were reported in Miami-Dade County, as Dak mentioned before, but nothing so far in Palm Beach County. So this is good news. It doesn't mean that it's not here or around the beans, but we haven't found anything on beans yet. So that just to summarize, the two maps I show here, these are from our pest and disease hotline from 2023 and 2024. This year, the infestations overall were not as bad as last year. So that's a good news. From Miami-Dade County, Homestead area, we got reports of Thrips parvispinus from various vegetables. The highest number was on peppers, as I think Doug mentioned, and some lower numbers on cucurbit, eggplants, and beans as well. It's established in Eastern Palm Beach County. This is what you can see here with all these dots. But this year, the situation was more under control, thanks to regular scouting, and proper identification. So last summer, Thrips parvispinus was collected from cowpea here close to Baglade from a cover crop. And this summer, we also collected parvispinus from our sentinel pepper plant on the station. So it's moving. It seems like it's moving, but so far, no report of parvispinus infestation from snap bean in Palm Beach County, and no report from West Coast either. So that's good. It doesn't mean that it's not there yet. It just wasn't detected yet. So keep an eye on it. We conducted a few insecticide evaluation trials on commercial pepper fields and cucumber fields. Why not on beans? Because the bean infestations were super low. Actually, we didn't have infestation on pepper on beans. So we had a cucumber and a pepper trial. So here we had 3 insecticide treatments. The first included 4 consecutive application of Radiant. The second had Radiant alternated with Transform and Vidate. And also the third represents a grower standard. including a rotation of tank mixed insecticide commonly used by the growers, and the water check at the end, and we replicated everything 4 times with an RBD design. So what so this was a small pot experiment, and was planted after an infested pepper crop. So this is how we got the infestation, and we conducted samples every 1 day after each application, and then 5 days after the last application. Collected 10 leaves and counted all the adults and larvae. So here, I have Thrips parvispinus adults, and on the other side, larvae numbers. And notice the difference on the Y axis. And I didn't put any statistics on it, but statistics generally supported the visual pattern. So for Thrips parvispinus adult across the 4 sampling dates, there was 40 to 70% decrease, really nice decrease. Relative to the untreated check. Treatment 1 and treatment 2 performed the best. Treatment 3 decreased the Thrips parvispinus adult numbers, but not as well as we expected. We are not sure why. We might have a little problem with the rain event, so that might contributed to it, but we are not sure. So larvae, they were Thrips parvispinus larvae, and most probably Florida flower thrips larvae, because that was the other dominant species in this test. And across sampling dates, we observed 65 to 85% of decrease in larva numbers relative to the check. So that was really great. Treatment 1 was the best again, with the treatment 2 and 3 having comparable efficacy. So again, a smaller trial, quickly, a small jalapeno trial after Thrips parvispinus infested pepper field. We planted this trial, and we had to initiate the applications pretty quickly because we got already infestation coming in. You can see here the tiny peppers, tiny thrips already infesting the pepper plants. So similar treatments as for the cucumber, except that we only had 3 applications 7 days apart. Again, I'm showing adults and larvae here. And for Thrips parvispinus adults, after each insecticide application, there was about 70% to 100% decrease, which is a great compared to the check. And then treatment 1 and treatment 2 seem to work somewhat better than treatment 3. Oops, sorry. And as far as the larvae, again, across all the sampling dates, if you look at the second chart, we observed 65 to 85% decrease in larva number relative to the check. Again, treatment 1 was the best, with treatment 2 and treatment 3 having comparable efficacy. This is just, I don't know why I'm going back. This is just a few pictures comparing the treatments with the check. This is how your pepper plant can look like if you don't treat it at the right time. So this is really scary. So early detection and action are very important. And just to sum up, again, should bean growers be concerned? Based on what we saw, based on all these surveys and everything, not as much as pepper growers, luckily. Pepper seemed to be the most impacted vegetable and most preferred host, as probably. If you grow any vegetable host, pepper, cucurbits, beans, monitoring is key. For management. Thrips parvispinus can survive and feed on beans, so beans can serve as a refuge. So pay attention to previously infested areas, your crops surrounded, be aware of potential infestation sources. Pay attention to nearby nurseries, know your neighbor and what your neighbor grow, and destroy pepper plants after last harvest if you grow pepper. If you suspect Thrips parvispinus, please contact us. We would love to go out and see if it's really Thrips parvispinus and help you with the management. So thank you for listening, and a huge thank you for Bad Nurse Farm because they made everything happen. And also to my colleagues out here. And with that, I conclude the IFAS presentations. And if you have any questions, I'm here all day, so we can catch up and because we are running a little late.